Construction of the first recombinant DNA. Construction. So we are discussing about biotechnology already, as you know that it. So in that, first topic is construction of first recombinant DNA. In this one, for the construction, the American geneticists Stanley Cohen and Herbert Boyer. Stanley Cohen and Herbert Boyer accomplished means finished construction of first recombinant DNA in 1972. That is first. So construction of first recombinant DNA. The word only saying the meaning, recombinant DNA. So the American genetist Stanley Cohen and Herbert Boyer used the accomplished the construction of first recombinant DNA in 1972. That is next one. Cohen and Boyer use restriction engines. Used restriction engines. That is E-core 1. So these two members use restriction engines. Restriction engines means cutting engine, DNA cutting engine. They use restriction engines. The restriction engines is also called E-core 1. So E-core 1, it is E-core 1. Restriction engine is also called E-core 1. This E-core 1 engine is collected from the Escherichia coli. Escherichia coli bacteria. It is coli bacteria. Escherichia bacteria. Okay, that's it. From the Escherichia coli bacteria, they isolated engine first time. That's why it is called E-core 1. E-core 1. E stands for Escherichia. In that Escherichia first letter is E. Core. C-O. Core. R. Rough strain. Rough strain or smooth strains are there. Rough strain. Roman letter 1. Restriction engine. Restriction endonuclease. Short form. Restriction REN. REN means restriction endonuclease engines. Restriction engine, short form will say restriction engines. Full form restriction endonuclease engine. Restriction endonuclease engine is collected from Escherichia coli bacteria. Restriction engines collected from Escherichia coli bacteria. Okay, that's all. So Cohen and Boyer used restriction engines. Called E-core 1. Which is collected from the Escherichia coli to cut the plasmid of salmonella. Okay, that Escherichia coli. From the Escherichia coli bacteria, restriction enzymes collected. Why they have collected? To cut the plasmid of Salmonella typhimurium bacteria. Salmonella full name, Salmonella typhimurium. From that bacterium, plasmid is isolated. That isolated plasmid needed to be cut. To cut that, they used restriction endonuclease enzyme. The restriction endonuclease enzyme is also called ECOR 1. ECOR 1. So this is the second point. Okay. So Cohen and Boyer used restriction endonuclease enzymes. It is called ECOR 1. ECOR 1 is collected from the Escherichia coli bacteria. Okay, to cut the plasmid of Salmonella typhimurium. So in the Salmonella typhimurium, plasmid is there. So to cut the plasmid, they used restriction endonuclease enzymes. So what is plasmid? What is plasmid? First PUC already I said plasmid. What is plasmid? Here one mark definition they will ask. Define plasmid. And autonomously replicating. An autonomously replicating circular extra chromosomal DNA. Autonomously replicating circular extra chromosomal DNA. Extra chromosomal DNA. Autonomously replicating circular extra chromosomal DNA. That is called plasmid. So that plasmid is present in bacteria, Salmonella typhimurium bacteria. Plasmid of Salmonella. So in the Salmonella bacterium, what is their plasmid? What is the definition of plasmid? The plasmid nature is autonomously itself replicating circular extra chromosomal DNA. That is called plasmid. Okay, so see here how that plasmid will be there. Like this, it is called plasmid. Using restriction endonuclease enzymes, they will cut the plasmid. How they will cut? See here. Like this, restriction endonuclease enzyme can cut. Okay, like this way. Okay, so this is complementary. See, it's PGG. Here, nitrogenous base sequence. It is a DNA, autonomously replicating circular extra chromosomal DNA. It is a DNA. This is DNA. So this DNA, this plasmid is cut with the help of ECOR 1. ECOR 1 will cut the plasmid DNA, leaving the nitrogenous bases. Complementary. Leaving the nitrogenous base complementary. Like this. Leaving the nitrogenous bases complementary. Okay, this is like this way. This is called plasmid. Okay, so initially it was circular. That they will isolate and we cut this plasmid at a specific sequence, at specific locations. Where is not cut? At the specific locations, those locations PGG base complementary, TCA complementary. Okay, this is called plasmid. Now, using restriction agents, they cut the plasmid DNA. That is one step. Next one. The antibiotic resistance gene. antibiotic the antibiotic resistance gene. Antibiotic resistance gene. The antibiotic resistance gene. The antibiotic resistance gene is isolated by cutting out of a piece of DNA. Cutting out of a piece of DNA which had resistance to antibiotics. Once again this point. Antibiotic resistance gene, that means desired gene. Desired gene. The plant or animal or any microorganism from them. Gene of our interest. Gene of our interest means desired gene. The plant which is growing superiorly or animal from them superior plants and or animals or microbes. The superior gene we liked. That gene we have to cut. That gene only called here desired gene. That desired gene is also called antibiotic resistance gene. Antibiotic resistance gene is isolated, separated by cutting out of a piece of DNA from that specific plant or animal or microbes. That DNA had resistance to the antibiotics. If we use any antibiotics, those antibiotics does not cause any harmful effects because those DNA had resistance to the antibiotics. So the antibiotic resistant gene means desired gene, superior gene from the plant or animal or any microorganisms. We have to collect. To collect that DNA, they will use restriction endonuclease. They will use restriction endonuclease. Okay, that is the step. Next step, cutting off a DNA in both the cases. In both the cases means what? Here one case, here one case. In both the cases they will use the cutting off a piece of DNA in both the cases. The same enzyme, restriction endonuclease or ECO1 use. Therefore, this enzyme is also called molecular scissors. Molecular scissors or biological scissors. Molecular scissors or biological scissors. Molecular scissors are biological scissors. That is the step. Okay. In both the cases, in this case, the DNA looks like this. The antibiotic resistance gene like this. This is antibiotic resistance gene. The antibiotic resistance gene or desired gene or gene of our interest what are alien DNA. Alien DNA or foreign DNA. There are number of names. Alien DNA or foreign DNA. Alien DNA or foreign DNA. That is this. So this is also complementary to this. It is T. Then here A, G, C, G, C, C, G, C, G, A. Okay. way they will use to cut the DNA. In such a way they will use to cut the plasmid DNA as well as foreign DNA or antibiotic resistance gene or gene of our interest or alien DNA. Okay, so the cutting of DNA in both the cases they will use the same restriction endonuclease. It is also called molecular scissors or biological scissors. So molecular scissors or biological scissors. That is the one step. Okay. Next point, next topic. So the piece of DNA having antibiotic resistant gene was then linked with plasmid DNA with the help of DNA ligase enzyme. The piece of DNA, the piece of DNA having antibiotic resistance gene. In this one, antibiotics resistance power is there. That's why it is called antibiotic resistance gene. A piece of DNA having antibiotic resistance gene was then linked with plasmid DNA with the help of enzyme. With the help of enzyme that is called DNA ligase. DNA ligase. This DNA ligase is also called binding enzyme. Molecular biology said that DNA ligase. With the help of this DNA ligase, this piece of DNA is linked. That now I will link. They will be fixed here. So when they will be fixed with the help of DNA ligase enzyme. So this is called autonomously replicating plasmid. As a result of binding of a piece of DNA into this plasmid, circular autonomously replicating new plasmid is formed. So as a result of binding this DNA into this plasmid with the help of DNA ligase, what formed? Autonomously replicating small circular plasmid DNA is formed. Now this is called recombinant DNA, recombinant DNA. The word itself signaling recombinant DNA. So when it is from different species, this is the gene of our interest, desired gene. It has a superior characters which is collected from either plant to animal, microbes, eukaryotic cell or prokaryotic cell, from wherever, but that gene is a superior gene. So that gene is isolated and inserted into the plasmid to form small circular DNA. Now this is called small circular DNA. Now this is called small circular DNA. Small circular DNA. So this is only called recombinant DNA. recombinant DNA. Recombined DNA. Okay, that's it. Next. So before inserting this is called plasmid. The plasmid acts as vector. Plasmid acts as vector. Vector means carrier. Okay, so if you want to transfer this into this is we presented in this one. This entire will be transferred into the another organism that is called E. coli bacteria. Now this entire recombinant DNA is transferred into the E. coli bacteria to multiply this DNA. To multiply this DNA. So that in the E. coli bacteria, foreign DNA is multiplied. If you wanted to multiply foreign DNA, here vector is needed. Directly taking DNA and inserting the E. coli bacteria manually does not possible. There is one vector is needed. If you wanted to go from here to mustang, something vector carrier is needed. Through that carrier we will go there. Directly we can go, but it's a difficult task for us. In the same way here also. Okay, a foreign DNA, this is. If you wanted to produce this number of copies, manually not possible. What we have to do, this is we have to insert the plasmid. Then the plasmid is transferred into E. coli bacteria. In the E. coli bacteria, these copies, multiple copies are formed. So that here plasmid acts as a vector. As they can transfer desired DNA. As they can transfer desired DNA. As they can transfer desired. Desired means attached DNA. This desired DNA only here attached. So that it can transfer attached DNA into another organism. From here to Hubli, we wanted to go means it is very difficult task. We want one carrier. Through carrier we will go. In the same way, this DNA we wanted to transfer into the E. coli bacteria. This is E. coli bacteria. This DNA directly if it is transferred to the E. coli bacteria, the process does not work out. Through vector, we can transfer into the E. coli bacteria. This is E. coli bacteria. So that here what we want, this is attached to plasmid. Then this plasmid is transferred into the E. coli bacteria. In the E. coli bacteria, the desired genes are multiplied. So that plasmid is required. Plasmid acts as a vector. As they can transfer attached DNA to it. Okay, that's as E. coli, Escherichia coli bacteria reproduce, as E. coli reproduce, it formed clone of DNA, as it formed clone of DNA. Clone of DNA. That means antibiotic number of DNA fragments. Like these fragments are cloned. Cloned means multiplied. Clone of DNA. Clone of DNA fragments. Clone of DNA fragments. Clone of DNA fragments. Clone of DNA fragments. That's that. Okay. So in the E. coli bacteria, as Escherichia coli reproduce, it formed a clone of DNA fragments. DNA fragments. That means antibiotic resistant gene. So in this one, again this plasmid will not appear. Number of DNA fragments. These are called clone. Clone of DNA fragments, that is antibiotic resistant genes. So like this way, alien DNA, foreign DNA is multiplied. The process of this process multiplying, the process of production of multiple copies of DNA fragments is called cloning. The process of production of multiple copies of DNA fragments, resultant DNA fragments is called cloning. On that day, on 6th chapter also says cloning of DNA. Methodologies, I said cloning. Cloning means what? Same copies production. Okay, so the process of production of multiple copies of desired DNA or antibiotic resistant gene is called cloning. So in the Escherichia coli bacteria, what happened? Replication of DNA. What DNA? This DNA. This DNA in the Escherichia coli bacteria replicated as E. coli bacteria performs replication of DNA and produces number of copies. The replication process, as replication process involves DNA polymerase. DNA polymerase enzyme. DNA polymerase enzyme is involved. Cloning. Okay, so the process of production of multiple copies of desired DNA is called cloning. Multiple copies of DNA is formed during replication of desired DNA in E. coli bacteria. So, Escherichia coli bacteria undergoes replication of desired DNA with the help of DNA polymerase and produce number of copies. This process is called cloning. The process of production of multiple copies of desired DNA with the help of DNA polymerase. With the help of DNA polymerase is known as cloning. Okay, that's all. This is the construction of the first recombinant DNA. Already we constructed. This is called recombinant DNA. This recombinant DNA is transferred into the E. coli bacteria. In the E. coli bacteria, it undergoes replication process with the help of DNA polymerase enzyme and produces multiple copies. Those multiple copies are desired gene. These are superior genes. These superior genes will be collected and inserted into the less superior plants. Inserted into the less superior plants. Those plants, either plants or animals, those plants are called genetically modified plants. Those animals are called genetically modified animals. This is the method of biotechnology. In biotechnology, the basic step this is. In horticulture university, here horticulture university is there. In the horticulture university, what I said now, this is practically they will do. Practically they will do. Okay, that's all. This is the explanation about the construction of the first recombinant DNA. So through this, number of desired DNA is produced. Number of desired DNA is produced. Those number of desired DNA, superior genes, those genes are collected from the E. coli bacteria and inserted into the plants or animals, etc. Those plants are called genetically modified plants. There are three basic steps are involved. There are three basic steps. There are three basic steps to make genetically modified organisms. Genetically modified organisms. The first step is identification of desired gene. Identification of desired gene. The first step, identification of desired gene. So that is the first step. Identification of desirable DNA with genes. Both are DNA, gene. In the DNA is made up of number of genes. Genes are made up of nitrogenous bases. Already said that. So first one is identification of desirable genes. Identification of DNA with the desirable gene. Second one, isolation of desirable DNA. First identified desired gene. That gene is identification. Sorry, isolation of identified or desirable gene and inserted into the organism, inserted into the host. Inserted into the host. Host means E. coli. Inserted into the host. That is the second step. In second step, identified desired gene. Sorry, isolation of desired gene and inserted into the host. Next, third step, maintenance of inserted DNA into the host organisms. So maintenance, this E. coli we have to take care of. Maintenance of host organism and introduced DNA. Maintenance of host organism. At that moment, what happens in this E. coli bacteria? Introduced DNA is multiplied. So those DNA we have collected and introduced into the progeny organisms. Progeny means what? Different organisms. Actually, progeny means what? Small organisms, offsprings, small plants or animals. So those plants or animals are called genetically modified organisms. So there are three basic steps are required to make genetically modified organisms. First one is identification of DNA with the desirable gene. Second one is that identified DNA needed to be isolated and have to be inserted into the host organism. And third one is maintenance of the host organism to make it multiple copies. When multiple copies are formed in this host, these copies we have to collect it and transfer into the different offspring plants or animals. So where we have transferred these desirable genes, those plants or animals are called genetically modified organisms. If we will insert gene into the plants, genetically modified plants. If we insert animal, genetically modified animals. Okay? That's all. Transgenic plants, transgenic animals. Genetically modified organisms. Genetically modified organisms. In case of plants, transgenic plants. In case of animals, transgenic animals. Genetically modified plants, genetically modified animals. This is the explanation about construction of the first recombinant DNA. It carries for 5 marks topic. Okay? That's all. Tools of recombinant DNA technology. So there are three basic tools are required for recombinant DNA technology. In those three basic tools, first one is restriction endonuclease. There are three basic tools are required for recombinant DNA technology. Among those, first one is enzymes. First one is enzymes. Second one is cloning vector. Third one is post. Enzymes are, I said today only. Cloning vector, plasmid, I said. Post, E. coli. These three are basic tools for recombinant DNA technology. Among those three, the first one is enzymes. Enzymes. So those enzymes are restriction endonuclease. Restriction endonuclease enzyme. So restriction endonuclease enzymes are belongs to two major classes. Actually, there are hundreds of restriction endonuclease enzymes are there. 900 different types of restriction endonuclease enzymes are there. Those 900 different types of restriction endonuclease enzymes are isolated from different types of 200 different strains of bacteria. So those restriction endonuclease enzymes are mainly belongs to two classes. One is exonuclease, another one is endonuclease. Exonuclease, another one is endonuclease. Exonuclease enzyme can cut the DNA at ends of DNA, end of the DNA, and the end region of the DNA. Whereas endonuclease can cut the DNA at middle of DNA, middle part of the DNA at the specific location. Keep in your mind, at the specific locations. At the specific locations only you can cut the DNA. Okay, so restriction endonuclease enzymes according to the situation. Okay, so if they will introduce exonuclease enzyme, it will cut the end region of the DNA. If they will introduce endonuclease, it will cut the middle of the DNA. Okay, so restriction endonuclease enzymes can cut the DNA, can cut the plasmid DNA as well as foreign DNA at the specific locations, so that they can leave a specific small strands. Those strands are called sticky ends. Sticky ends. What are those sticky ends? Already said that. These are called disease, 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 sticky ends with complementary. Disease, sticky ends with complementary sequence. Sticky ends with complementary sequence. Sticky ends with complementary. In the same way, even in plasmid also. Already I said. This is the sticky end. This is sticky end. This is sticky end. Sticky ends. Okay? So restriction endonuclease can cut the plasmid DNA as well as foreign DNA so that it can leave small strands. Those strands are called sticky ends with complementary with each other. Therefore, these enzymes are called molecular scissors or biological scissors.